The next speaker is Jeff Shaman.
Hi, I will talk about repeat infections, in fact, and I'm going to start sharing here. Okay, so I'm going to talk about this work that we did with a cohort doing active sampling in New York City, looking at a variety of respiratory viruses. And specifically what we found looking at some of the betacoronaviruses that may be of relevance to contextualize some of the things we're going to have to contend with respect to repeat infections for SARS-CoV-2. So as I'm sure all of you know, typically surveillance for viruses, particularly respiratory viruses, really only gives you a little bit of information. We have a broader population. We have a swath of people who are infected. Only a fraction of them are typically sick enough to actually seek care, and then only a portion of those that seek care are tested and actually virologically confirmed. So we're only getting a very limited perspective of what's going on in the broader community. When we look at estimates of what the total burden of a respiratory infection is at population scale, typically there are three methods that are used. Serological testing. This is usually done retrospectively, though obviously in this context we may start seeing some active rollout of some of the techniques just spoken about are ramped up and brought to scale. But generally this is retrospective and it's indirect. We're looking at past indicators of an actual infection. Then you have just sort of statistical inference methods where you're going to build some multiplier model to starting from measurable outcomes and develop some probability distributions to actually estimate what has been going on. Often this is based on self-reported ILI and some known indicators of overall prevalence of the virus based on historical ILI conditions. This is often used, but the estimates you get from this are widely varying, which suggests that it's subject to a lot of inaccuracy. And the last one is to try to more directly measure it, and that's with a longitudinal study where you're actually going out into the community and actively sampling portions of the population to see what's going on broadly in that community. This is something that we actually did. We set up a cohort over the 2016-17 and through the end of the 2017-18 cold and flu seasons here in New York. We enrolled 214 individuals into this cohort, and we asked each of them to keep a daily diary of cold and flu symptoms from a common cold questionnaire: cough, sore throat, runny nose, watery eyes, chills, etc. They'd rate it on a Likert scale, none, mild, moderate, severe, and they'd enter it on a daily basis over their phone electronically into our database. They'd also tell us at the same time on a daily basis whether they had taken medicine, stayed home from school or work, or sought medical attention because of their respiratory conditions, their symptoms. We then, once a week, irrespective of how they felt, we would swab them, take a nasopharyngeal swab, and we would test it using a respiratory viral panel assay for 18 different respiratory viruses. So this is showing you some of the results from that. We had participants from 2 daycares. These were some very young children, some of their siblings and their parents. We also had employees, people who work at CUMC. We also sampled in a pediatric and an adult ED among healthcare workers there, and we also sampled in a high school. So it was a range of people that we got. We did not have anybody over 65, I should note, in the entire cohort. What we have is showing you here is a record of the number of viral infectious episodes in that column labeled episodes. It's a group of consecutive weekly specimens from a given individual that were positive for the same virus. We did allow a 1-week gap to account for false negatives or temporary low shedding. And what you see are some of the things you might recognize broadly. There's a lot of human rhinovirus infections, and there's also a lot of endemic coronavirus infections that were recorded there. MA says is indicating medical attention, and that's the number of episodes of these identified episodes for which the individual sought medical attention. And you can see that coronavirus comes out at the bottom of it. The probability of seeking medical attention given infection with a coronavirus, and there were 4 different coronaviruses typed, so we had them individually, was only 4%. There were only 6 individuals who bothered to go and seek medical attention. So it's typically very clinically mild. You can then go further to right and you can see how often people stayed home. And again, coronavirus is the most mild of the infections that we documented. And then lastly, taking medicines. You can see the more severe ones are both influenza and human metapneumovirus, as would be expected. Now, within this cohort, we have 12 instances of individuals having more than one infection by the same coronavirus type within the span of a year. Most of them are the betacoronaviruses. There's one with an incredibly low signal that we suspect may be spurious, that is 229E, which is an alpha coronavirus, but OC43 and HKU1 are the betacoronaviruses here. We are in the process of sequencing all these because we want to confirm them. A few of them have been sequenced, and we have particular concerns that the OC43 signals that you're seeing here, this is not really a quantitative PCR, but it does produce a quantitative signal. It's just not reliable. It's a GenMark respiratory viral panel system. They call the cutoff for OC43 this measure of 25 nanoangstroms per millimeter squared. This is electrical conduit signal that ultimately comes out of the test. But we're a little suspicious that that may be too low, because as you can see in this orange right here, I don't know if you can see me moving over with a cursor here, those two we did not pick up when we sequenced it, where the ones in green that were sequenced that are above 100 were in fact verified upon sequencing. So we're sequencing all the rest of them. But be that as it may, you can see the age distribution there. A lot of them are very young, but we do have some older people, 25, 31, and 35, as well as a 9-year-old who had repeat infections. We also note that there are two clusters of time scale at which the repeat infections occur. Basically 4 weeks to 2 months. And in those instances, those are the ones where we have the weaker signals. So those may be indeed spurious. Maybe something about the virus coming up again. It may be a lag, or it may just be, poor sensitivity and specificity in this case of the test. Of greater security, we have, I'm sorry?
It's your 1 minute mark.
Thank you, Andre. Of greater concern is the fact that we see these clusters at 8, 10, and 11 months, where we're seeing repeat infections that are very well separated in time, have very strong signals. The symptom scores, which go from 0 to 27, well, no, actually above that, their aggregated symptom scores over multiple days show that sometimes people experience more serious symptoms upon repeat infection at 8 to 11 months than others, and sometimes they go down. But what we're seeing here is clear evidence of repeated infection taking place by the same type. We've done some survival analysis on this to actually look at what the probability of this means, just to model it and see what the fraction is. OC43 is there in the red, HKU1 is there in the black. Obviously, we don't have very many HKU1 samples there, but it's showing a fairly high probability of reinfection. Now, this assumes that survival is independent of time of enrollment. It doesn't factor in seasonality of disease, of the virus itself, which may exist, and a number of other factors. But we're seeing substantial evidence of reinfection at a very short time scales, which is quite concerning. This is another shot of one where we excluded all signals less than that 100 nanoangstroms per millimeter squared. We're still getting something that looks very similar at that year mark of a huge drop in basically immune protection for reasons that we don't quite know yet. This just shows some similar survival analysis plots worked up from the start of it as your onset time to see when you get an infection. And it sort of trails at a similar fashion, but since I'm running short of time, I won't go in on it. This is work that Marta Galante and my team has been leading. I want to stress that she's been doing a lot of the work on it, and we are going to be sequencing it. But I think one of the key things here is it's suggesting either we have a situation of waning immunity or immune escape. We're going to be able to look at the immune escape a little bit with this by doing the sequencing and seeing what the viral sequence actually looks like. The waning immunity is something that I believe I've heard some talks last week. I wasn't able to be on much of the time, that were suggestive that there may be some issues associated with that. And certainly we have evidence in the literature, though it's not large, that both SARS-1, which is most closely related, sees waning immunity of antibodies within the span of about a year, though the memory T cells seem to persist. And as well as that other coronaviruses, endemic coronaviruses, there are a few papers that are looking at various challenge studies and otherwise that there is waning immunity within these coronaviruses. So I will stop there.
Thank you, Jeff. If you can please turn off your slides. So there's one, Darcy. Go ahead, Darcy.
There's pigs have been studied for a long time in terms of respiratory and enteric coronaviruses, and Linda Saeed has thought that in fact the mucosal side of the disease makes the development of long-lasting immunity very difficult and has hindered vaccine development. I wondered if you could comment on that.
You know, I'm not aware of those studies and I really should look into it, but I mean, that sounds like one putative hypothesis. I mean, given the evidence that I have seen to date from those, the limited work on SARS-1 and looking at it in humans for the endemic coronaviruses, my suspicion is this relates more to rapidly waning immunity than it has to do with antigenic drift and immune escape. But this is something that obviously has implications for as we try to develop a vaccine, how we roll out a vaccine, whether repeat doses of a vaccine are going to need to be delivered just to keep people boosted, and whether using serological assays to let people back out and going to work, even though, to be perfectly honest, you know, the number of people who are infected right now is a drop in the bucket, and if we get a handle on it within the next couple months, there's still going to be an enormous fraction of people who are susceptible. But using those serological assays, we may have to do them repeatedly to make sure people are up to date in knowing what their immune profile is against this pathogen.
Thank you, Jeff. So I have a question. So when you see repeat infection, one could be sort of either non-mature immune system, and so you know, that may account for the younger children. Another one may be loss of memory B cells, but also there's the possibility of mutations in the spike and HT proteins, the antigenic proteins in the virus. So do you have a sense of which, you know, how this thing breaks? For instance, do you see more, you know, fewer mutations in the younger population versus the older ones? You know, have you been able to look at that kind of breakdown?
No, not yet, as a matter of fact. So you know, we pulled these out and we're looking at them. We hadn't looked specifically at the coronaviruses, but we pulled them out and we just done this analysis at this point, and we're literally at the point now, we prepped all the samples to set them off to sequencing. So we want to look at that so that we can look specifically to see how the virus has changed and whether we're seeing some evidence of antigenic drift and immune escape by the virus versus something that, whether or not we could rule that out and look at and say this is something more related to host immunity.
Another question from Sinead Morris.
Hi, can you hear me?
Yep.
Hi. So I just had a question about cross immunity. So for example, I was wondering if a participant is infected with, for example, HKU1, do you see any evidence that they are less likely to be infected with OC43 in the following?
We haven't looked at that yet, and that's a line of inquiry that we want to pursue subsequently from this. But it's a great question, whether or not there's really any evidence from this cohort of that kind of cross protection. But we haven't examined it yet explicitly.
Okay, thank you.